Cryo-electron microscopy sort of blends two components. Cryo basically means we're working at very, very cold temperatures. In this case, at or below sort of liquid nitrogen temperatures. We do that to preserve biological specimens so that they're not damaged when they're put into the microscope and imaged. The microscope itself, it works very similar in many ways to a light microscope, except that it uses electrons instead of photons to image samples. So CryoEM basically is the merging of an electron microscope with cryo to preserve biological specimens. The data that a cryo-electron microscope captures is quantitative in that it basically gives us a picture of the particles we're looking at. We image a sample with electrons, and then we actually count each individual electron as it impinges upon a detector. And depending on where those electrons fall on the detector, they eventually build up this picture of the molecule that we happen to be looking at. In terms of reproducibility, one should be able to look at the same molecule with the same type of instrument, no matter where you are, and get essentially the same type of result from that. We can hopefully glean some kinds of insights into physical mechanism and biological function.
Another technique we've used in the past is a technique that utilizes X-rays. It's called X-ray crystallography. The challenge with that technique is that in order to use that method, you need a crystal of what you're interested in. And getting that crystal is very difficult, and it's very hard to get biological molecules to form a crystal. CryoEM has really revolutionized structural biology because it's provided us with a new way to get structures of these biological molecules and then understand how they work.
My lab works on the structure and function of chromatin, which is the material in which genomes are organized inside the cell. And we're interested in understanding how chromatin is organized because we want to know how the information encoded in the genome is utilized in the cell to carry out its many activities, especially during differentiation and development into the many tissues of an organism. And to do this, we use primarily the tools of biochemistry and genomics and molecular genetics to identify regulators of gene expression and to purify these proteins and study their activities. All manner of diseases are caused by defects in the structure and the function of chromatin, including things like cancer and other diseases of cellular metabolism, developmental diseases, and so on. So it's really fundamental to all of biology and biomedicine. When chromatin remodeling becomes dysfunctional, there are consequences. Sometimes there are catastrophic consequences. And so these very large enzymes are prime drug targets. They are potentially targets in which you can either activate an enzyme which is normally not working, or find a way to suppress an enzyme that is overactive. And in order to really either do drug design or screen for drugs in an unbiased manner, it really is almost essential to have knowledge of the atomic structure of these protein complexes. And so that's the reason why CryoEM is so important to us, is that it fulfills both the thirst for fundamental knowledge, but also allows us to then think for practical applications to be able to manipulate these enzymes for patient care.
One of the really exciting things is that, let's say, a patient that has a certain disease, what we will be able to do and what's currently in place here at the Beckman Center is we can take patient samples from our clinical counterparts and actually specifically understand what is going on with a protein that may be causing their disease. And oftentimes with how high throughput CryoEM is now, is that this may be something that is actually even more informative and more accessible and quicker than sequencing, for example. Things like, let's say, SARS-CoV-2 mutations. We can develop a platform where we can take these samples from patients and in real time solve the structures of these viral particles that would then allow us to delineate these mutations and how they may actually be evading vaccines.
My lab studies the CRISPR system. This is a system that's found in many single-celled organisms, for instance, bacteria. It is an immune system for the bacteria. So bacteria, just like humans, have evolved many immune systems to protect against virus infection. And so the CRISPR system is one of those systems that we study. It's particularly significant because we've been able to leverage the CRISPR systems as tools in medicine and in research. We get this snapshot of a CRISPR complex bound to a DNA sequence that really we don't want it to bind to. And what the hope is that we can analyze these sequences, and we're collecting lots and lots of these views that will understand the rules of how it accommodates these variations. And the sort of long-term goal is, can we change the CRISPR complexes in some way that stops binding to these variant sequences and makes the system more specific? A sort of complementary technique to CryoEM would be something called X-ray crystallography. So my lab, when it first started at Hopkins, started by using X-ray crystallography. And the first structure of a CRISPR complex we determined took us 5 years to do, from sort of start to finish. Doing a similar project now, we've completed it in about 6 months. The technology is much easier for these kind of problems. New tools and new techniques let us ask new questions. We can go deeper into a problem because we have technology that lets us, for instance, in this case, visualize something we couldn't visualize before. Or they make a process that was very difficult and time-consuming much easier and quicker, so we can progress quicker in our understanding of a given biological problem.
The ability of science to make new advances is really only as good as its tools are to do precision types of measurements. And that is why technology is such a driving force, and why, as technology continues to improve, we keep seeing more significant scientific advancements. The Beckman Foundation has a legacy of supporting science broadly, both in terms of young investigator science, but also in terms of instrumentation and instrumentation development. That type of support is really critical in the sciences. As we move through time and instrumentation becomes more complex, it also becomes that much more expensive. And so securing large amounts of funds to be able to acquire instrumentation, to be able to use it, maintain it, is quite a challenge. And the Beckman Foundation has really been a pioneer in supporting science for science's sake. And at Hopkins, that's also in our DNA. Hopkins is really built on the idea of conducting scientific research in the knowledge that information ultimately is a good thing and it's powerful and you can do things with it. And down the road, that information can be translated. We can do practical kinds of things with our discoveries, but first you have to learn it. And that's what the Beckman Foundation allows us to do, is to acquire the instrumentation necessary to really make discoveries that haven't been made before. The initial award that was made to us by the Beckman Foundation has really been pivotal for building cryo-electron microscopy and 21st century macromolecular imaging here at Hopkins. With the money that we received from the Beckman that went towards the acquisition of our first electron microscope, Titan Krios, we have since been able to leverage that into hiring an assistant professor doing very exciting work in neurobiology who works with cryo-electron microscopy. We've just made another hire at an assistant professor level on a person who's working on viral structures, also using electron microscopy. With that initial investment, we've been further able now to acquire another microscope through the Howard Hughes Medical Institute for basically conducting screening studies that allow us to use our existing microscope more efficiently. And then most recently, we've been able to acquire yet another instrument which is useful for preparing samples so that you can look at molecules inside cells. It's for a technique called electron tomography. And none of this would have been possible without that initial seed funding from the Beckman Foundation. Dr. Beckman had a well-known reputation and legacy for being a pioneer in instrumentation development. And he made no secret of his viewpoint that instrumentation is a major driver of scientific discovery. And at Johns Hopkins, we completely buy into that perspective. We also believe that in order to make discoveries, you really need to be able to do measurements of all different kinds of measurements of all different sorts, depending on the field that one is in. But we'd like to think that by conducting the science that we are with the instrumentations that we use, many of which were developed by Dr. Beckman, that we are continuing in his legacy.